Good morning, and welcome again to the Summer Professional Development Institute Speaker Series. I'm Mike Mills, Vice President of E-Learning, Innovation, and Teaching Excellence at Montgomery College, and it's my pleasure to have Elite sponsor this week's speaker series. Today, we're excited to have Dr. Collins Jones from Montgomery College join us. He's the second speaker in the speaker series. Later today, Dr. John Byers from the University of Maryland Global Campus will present, and then tomorrow, we will wrap up the speaker series with Dr. Thomas Bailey, also from UNGC. Yesterday, we had Dr. Katie Linder join us from Kansas State University. Her presentation and slides are on the Summer Professional Development Institute website, which we'll put into the chat box so that you'll have that link. Colin's slides will also be made available on the website later today. So with that, let me turn it over to my colleague, Shinta Hernandez.
Good morning, Montgomery College. Wow, I am so excited to see this many people on the Zoom presentation. Thank you so much for being here for the Professional Week Speaker Series. I am Shinta Hernandez, Department Chair of Sociology, Anthropology, and Criminal Justice at the Rockville and Tacoma Park Silver Spring campuses. And I am proud to introduce our second presenter during Professional Week 2020. Please welcome one of our very own, Dr. Collins Jones. He is the coordinator of the Biotechnology Program here at MC. He works very closely with a number of Maryland biotech companies to develop and maintain curricula for the Biotechnology Program, including Bioreliance Sigma Millipore, Astra Zeneca, Lonza, and GlaxoSmith Kline. Dr. Jones also serves as a member of the Montgomery County Life Sciences Business Incubator Review Board, as well as being on the advisory board for the Master's Program in Biotechnology at the Johns Hopkins University and other local higher ed institutions. He spent 10 years at the National Institutes of Health as a research scientist, and he earned his BS in biochemistry with a mathematics minor from Albright College and his PhD in biochemistry from the University of Maryland. This morning, you will hear Dr. Jones' presentation entitled, What You Should Know About COVID-19. But before we get started, let me just give everyone a rundown of the logistics. While Dr. Jones presents, we will monitor the chat box. So you can type your question in, but when you do that, be sure that the chat box is set to everyone so that we can read your questions, and we will try our best to answer at that very moment. We'll also allocate the last 15 minutes or so for a Q&A session, so you'll have an opportunity to ask questions then. Dr. Jones's slides will be made available, as Dr. Mills pointed out, on the Summer Professional Development Institute later on. So without further ado, let me introduce to you again, Dr. Collins Jones.
Good morning, everyone. Thank you for attending. Shinta and Michael, thank you, and thank you for dealing with my being administratively challenged to get this up and running, but my other administrators are well aware of this. But we are here and ready to go. So I've put this presentation together, entitled it, What You Should Know About COVID-19, and I've sort of taken the approach that this is science for non-scientists. So if there's hardcore scientists out there, please email me your questions later, but really what I want to do is make this more accessible to a general audience, and I'm going to lead you through several things: an overview of the virus, how it's transmitted, how we detect it, what your immune system does once you're infected, the therapies that we use to treat it, and a little bit about how we create vaccines for this, okay? But first, I want to start just with a disclaimer. I updated this last night to include the Moderna vaccine results that were on the news yesterday. This is for informational purposes. As all of you know, the information about COVID-19 is changing on a daily basis, and there's lots of conflicting points of view about all the different aspects of this pandemic. So this is what I know to the best of my ability, and I have compiled this from looking at scientific journals on the web and from government websites, CDC, NIH, all those sorts of things, as well as other countries' websites, just to get a broad view of this. So if there's anything that triggers you to take some action before you do it, right, consult an official website, CDC, NIH, the Montgomery County COVID websites. They all have up-to-date, actionable information. All right, so are you guys ready? Let's get started. What is COVID? We all have heard about it. We kind of know what it is, but it is a virus, and it is a member of the coronavirus family, and it was first identified in the early 1960s. Coronaviruses cause colds and upper respiratory infections. So we've all been exposed to different coronaviruses than the one causing the pandemic, but these are a source of the common cold. And they infect not just humans, but animals, cows, pigs, turkeys, bats, sometimes dogs and cats. The other well-known members of the COVID family are MERS and SARS, Middle East Respiratory Syndrome and acute respiratory syndrome. Okay, so this is a picture of COVID, and it's called COVID because it has a corona. All right, COVID was first identified by June Almeida in England. All right, and this is a picture of June with her two colleagues. And what's interesting is June had no formal degree. She left school at the age of 16. She couldn't afford to go to college, but she did get a job as a lab tech in England, and then she later moved to Canada because England didn't recognize people without degrees. And she passed away recently, but she ended her career being a yoga instructor of all things, which is kind of cool, okay? And then we just have a picture. I'm not really going to go into the origin of COVID because there's lots of conflicting points of view, but the dominant theory is that bats are a reservoir for the COVID virus, along with other viruses, including Ebola and things like that. The bats then transmit this virus to either other animals or to humans if we consume it as a food. And then what makes this scary is that we have human-to-human transmission. So, and this is the whole idea of the marketplace and things like that. The flu virus also is transmitted similarly from animal to human, and then humans go to a marketplace. They exchange the virus with other humans who then go to the big city and infect other humans in the big city. And then those folks get on airplanes and they infect people worldwide. And that is essentially how we get a pandemic. All right, so COVID is the coronavirus that causes the coronavirus disease. So they've actually named this the coronavirus 19 disease, all right? It was identified by examining cases of atypical pneumonia in Wuhan. And atypical pneumonia is sometimes known as walking pneumonia. So while it did cause fatalities, it did cause death. Lots of people, as you know, who get COVID have mild to moderate symptoms, and pneumonia is just fluid in your lungs, and so they're able to walk around. COVID, the name of COVID is still evolving, so you may now see COVID-19 named as SARS-CoV-2, and there's also a SARS-CoV-1. COVID infects pretty much every cell in your body, but the lung cells are the primary target. Other cells include your kidney, your heart, your liver, and intestines. Okay, so this is an electron micrograph of the virus, which are the little yellow particles here coating a cell that's about to die. All right, and the cell is already infected with virus particles. But what I want you to notice about this is how many virus particles there are per cell and the relative size of the virus compared to the size of the cell. And then when the cell dies, it bursts open and releases all these virus particles, which then scatter to infect other cells in your body. So this is an electron micrograph of the cells. This is taken almost at the molecular level, but not quite. Okay. Now, before we continue, this is your first science lesson. We need to understand a little bit about how DNA works. All right. So DNA, we've all heard of DNA, deoxyribonucleic acid. It contains all the blueprints or the recipes to make proteins in the cell. Specific blueprints or recipes are called genes, all right? And a gene is just a sequence of nucleotides, which are the chemicals that make up DNA. And that is the point I want to make for this, is that DNA is just a chemical. It is not alive. And the chemistry of DNA is the same for every living organism on the planet Earth, including viruses, which some of my fellow faculty members consider viruses to be alive. Others, such as myself, do not consider viruses to be alive. But the idea here is that DNA can work in every single cell. So DNA is made up of nucleotides. A specific sequence, a specific piece of DNA that contains the information to make a protein is referred to as a gene. Okay? And so these are the nucleotides, the A's, G's, T's, and C's. And if we had more time, I'd show you the chemical structure, and we'd all be happy that there's no quiz or exam at the end of this presentation. Okay? But these letters here are the nucleotides, the chemicals that make up DNA, and the order of the letters, okay, G, G, A, A, they determine the recipe, they determine the blueprint, the information to make a protein. To make a protein, we copy these letters into another molecule called RNA. This is a copy of the gene, right? And again, that's another set of letters called nucleotides. Those nucleotides are then translated to make a protein, and a protein is made of chemicals called amino acids. And this is about as scientific as we're going to get for the talk, so you guys can kind of relax after the slide. But those amino acids then fold to make a protein, right? And proteins have specific shapes. And the shape of the protein determines what the protein will do in your cell, or what the virus proteins will do. And so this sort of sets the stage as to why we study all these different parts of the virus. Different proteins have different shapes. So the shape of the protein determines its function. The shape of the protein is determined by its DNA sequence. If I change the DNA sequence, the shape of the protein changes and its function will change. All right, so a virus, basic virology here. A virus is just a package of genetic material, DNA or RNA. Envelope viruses are coated with what we call a lipid membrane. So the viruses actually steal part of your cell's membrane and coat themselves with it, and it helps them hide from your immune system. So some people have called envelope viruses nano grease balls. A single virus particle is called a virion. Viruses cannot reproduce by themselves. That's why many of us do not consider viruses to be alive. But viruses can infect other cells, and these are referred to as the host cells. And it is believed that for every cell on the planet, there is a different virus, at least one different virus able to infect those cells. So viruses can infect bacteria, they can infect plants, and they can infect animals, including humans. Right? Once the virus is in the cell, it wants to replicate, it wants to make copies of itself, and it takes over the cell and uses the protein synthesizing machinery of the cell to copy itself and to make proteins for itself. And interestingly, some viruses can actually infect other viruses. So we have a schematic here of a virus. We have the membrane coat. There are proteins that are in the surface of the virus, so these are sticking out and these are exposed to the other cells in your body, and then inside we have the genetic material, and then there are proteins inside the virus to protect this genetic material. So this is a really compact little package here. So then the question is, how big is a virus compared to other things, right? And this is going to be important when we talk about masks and protection and things like that. So right there is our virus. It is in the 10 nanometer to 100 nanometer size. So this is not something you can see with your eye. You can only see this through a special instrument called an electron microscope, right? And you know, this is not to scale, this picture. So I've included a picture where a little more to scale, not exactly to scale, but this would be the, if you will, the relative size of an animal cell, a bacteria cell, and then a virus particle. And this is still not to scale. The bacteria cell and the virus particles are smaller than what I've represented here, but you wouldn't really be able to easily see it. So the idea, though, is that viruses are very small particles, right? Lots of viruses can fit inside a single cell. Okay, so COVID is what we call an RNA virus. The genetic material for COVID is RNA, not DNA. It is enveloped. The surface has a net negative charge. So the surface of the virus, the proteins on the surface, are negatively charged. Think static electricity. Okay, and this is going to have some implications for new masks that people are designing to protect you from the virus. Right? The COVID virus is a fairly large virus in terms of how much RNA it has, its genome. It has about 10 to 20 proteins, but the ones that we care about are the proteins that are on the surface of the virus. So this is a schematic of the virus, and you can see these little knobby things sticking up. How's that for a scientific description? These little knobby things sticking up are the proteins on the surface of the virus. And later we're going to talk about what each of these proteins are and what they do. All right. So for COVID to replicate, once it enters the cell, it converts its RNA to DNA, and this process is called reverse transcription, and that offers a target for drugs that we're going to talk about later for therapeutics. Once reverse transcription has occurred, once the viral RNA has been converted into DNA inside the cell, then we can go back and do the DNA to RNA to protein that we showed in the preceding slide. And then this is a little more basic schematic of the virus. Here we have the surface proteins, the envelope protein, the spike protein, which everybody is talking about, the nucleocapsid proteins, which basically protect the RNA inside the virus, and then the membrane protein, which helps hold the virus together. All right, now this is really scientific here. This is just to appease some of my science colleagues, but the genome, the sequence of the RNA in COVID has been worked out. And I want you guys, as we go through this, I want you to think about this. This virus emerged in late December 2019. Here we are almost 6 months later, and we have this whole wealth of information about a virus. We're making therapeutic drugs to treat the virus. We're making a vaccine. This really, the speed at which we've gained an understanding of COVID and been able to synthesize to get drug candidates and vaccine candidates is unprecedented. It's really quite amazing. Because if you remember, from the time that people identified AIDS to the time that they developed therapies for AIDS, it was 10 years. 10 years. And here we are 5 months later, and we're starting to bring therapies on the market. Okay. All right. So I'm not going to dwell on this, but I just wanted to show you that this is complex, but we are, every day there's new knowledge coming up on how COVID-19 functions. So let's go to transmission. All right. How is COVID-19 transmitted? Person to person. So the next couple of slides, we're going to see a lot of sneezing here. All right, so it's transmitted through sneeze droplets, person to person, and these are called respiratory droplets. And people are studying whether or not aerosols can also transmit the virus, which are smaller droplets than the respiratory droplets. When somebody sneezes, these droplets can be inhaled through your nose, through your mouth, or they could get in your eyes, right? And the droplets can fall on a surface, and if you touch the surface where the droplets have fallen down, and then you rub your face, then you can transmit the virus to yourself. All right. So MIT studied sneezing quite a bit. They took pictures of people sneezing to see exactly the trajectory of the sneeze particles. Okay. And what we see here is the study suggests that droplets of various sizes are trapped in a turbulent gas, allowing them to travel up to 26 feet. But the large droplets, the ones that are going to contain the most virus, they're going to fall within 6 feet. And that's where we get our social distancing from. So it's the study of the patterns of how people sneeze and where those sneeze droplets and particles go. Right. Now, the evidence is not in about aerosols in terms of COVID, but for other diseases, they have measured this. So Ebola the droplets which will transmit Ebola basically drop to the surface within 3 feet, but measles transmit out to 6 feet and beyond. All right, and measles is, by the way, one of the most infectious diseases on the planet. Okay. All right. So droplets are about the width of a human hair. Okay. One sneeze can eject roughly 40,000 droplets. Just coughing or talking, they produce about the same number of droplets, 1,000 to 3,000 droplets per minute. Each droplet will contain about 500 to 1,000 virus particles. So when someone is sneezing, it's not just the liquid that's coming out, that those liquid droplets are containing little particles in them, okay? So again, these droplets can go up your nose, down your throat, into your eyes, and they can deposit on surfaces. So here's our little droplet surface. The droplet is full of virus particles. So when you see people sneezing, this is what you guys are going to think from now on. Even though the vapor, the liquid may evaporate, the virus particles are left on the surface, right? And that brings us to the question of how do we protect ourselves? So the first way that you do this is you do physical distancing. I like physical distancing instead of social distancing. It's just me, but I like to be social, so I'd rather just say I'm physically being distant from you, but we could still socialize, all right? So based on our sneeze studies, right, and this is a physics problem, if you will, all right, Based on our sneeze studies, the recommended distance is 6 feet because within 6 feet, most of the droplets containing the virus will drop to the ground. And one thing that you have to think about is it's not just whether you get the virus, it's also the dose of virus. So people have not established yet what the minimum number of virus particles are, of COVID-19 particles are, that you need to inhale to actually get infected, right? So anthrax is a very small number of particles. People have studied that, but the data is not out on how many COVID-19 particles you have to inhale, you have to get inside of you before you get sick, all right? So aerosols, though, can travel up to 25 feet. That's why people are starting to study aerosol transmission as opposed to droplet transmission of COVID. And the aerosol particles, because they're so small, they can actually float around the room for 2 to 3 hours, okay? So social distancing, right? We have social distance warriors. I like this when I saw this on the internet. So you can see some people, and I've actually saw this at the Target the other day, about 3 or 4 weeks ago. Somebody actually had a tape measure. This is not that person. I did not take a picture of that person, but somebody actually had a tape measure to try to keep people away from them. In Germany, one of the cafes has decided that people are going to put tubers on their head to make sure that they all stay different distances away from each other. But mostly what you're seeing in the stores now is that we've measured out 6 feet and they've put little markers to tell you where to stand, okay? So physical distancing, here's an infected person. These are 6 feet distances between each person. So this should work because when this person coughs, sneezes, or talks, all the droplets should hit the ground before they reach any of the other people. But if you don't social distance, okay, then you're remembering that these droplets are coming here and you go close to somebody to talk and you're now within range of the droplets, you can get infected. So when that happens, I just leave, okay? So how do we prevent this? We protect ourselves with barriers, all right? So you're on one side of the barrier, the droplets are on the other side of the barrier. So if social distancing isn't working, which it wasn't for a lot of people, then we go to using personal protective equipment. So we want to cover ourselves up and have our own barrier. So this is a little bit extreme. Here's the social distance line, but this person has completely covered themselves and has their own independent airway. Most of us are not going to go to that extreme. What we are going to do instead is we're going to wear face masks. And there are 2 basic types of face masks. There's the surgical mask, which offers one-way protection. Basically, it catches your sneeze, it catches the droplets that come out of your mask, that come out of, that come out of your mouth when you cough, sneeze, or talk, okay? It is loose-fitting, it's what we use for the general population. They're relatively cheap. And then the masks that are in short supply are the N95 masks. And these protect both you and the people around you. So it will capture what's ever coming out of your nose and mouth, but what's coming at you will also be captured, right? And this is sort of a material science type of thing. And so what we have here is just a comparison. Tea cloth, this is homemade. This actually came from a website in India where they looked at how many particles get through the mask, all right? So protecting yourself, all right? Surgical mask, about 25 particles. If somebody coughs at you, about 25 particles will get through the mask, all right? If you are coughing outwards, you're not going to leak that many particles out, okay? So the idea is that if 2 people are wearing the mask, then both masks protect each other. Making the masks is, again, a material sort of issue. So this is a picture, this is another electron microscope picture of the fibers that make up a surgical mask. This is a cotton t-shirt, this is a pillowcase, this is a paper towel. My neighbor actually made her mask out of a vacuum bag because she realized that it traps dust particles. And then this is just tissue. And so what you can see is how fine the surgical mask particles are. And when they make a surgical mask, they actually have layers of this particle. They're blown one over another. So by the time you get all these tangles stacked up in 4 to 6 layers, virtually nothing gets through. Whereas in the rest of these, there's lots of little holes here that you can see where the virus particles could slip through. Okay, so that's a little bit about the science of masks and why we wear them. So some do's and don'ts of wearing masks. We've all seen this, but I'm not claiming to be an expert on putting masks on. There are plenty of websites, but some of the things that you see people doing, they leave their nose exposed. Guess what? The virus particle will go right up their nose, all right? They leave their chin exposed, right? This is not a tight fit, so the virus particle can go under the mask and up into their mouth or nose. And then we have the loose fitting masks, right? And so the sides are open and the virus particle can go in through the side. So it is really important when you put a mask on, it's not just putting the mask on, it's putting it on properly and wearing it. All right, new technology, the electroceutical mask. This is not on the market yet, but this is in development. If you guys remembered at the beginning of the talk, I mentioned that the virus has a charge on its surface, it's negatively charged. So scientists are learning to take these fibers that make up the mask and give them an electrical charge. And that way, when the virus comes onto the mask, one of two things happens depending on their approach. The virus is negatively charged. So if we make the mask negatively charged, it will repel the virus, right? Remember this from elementary school, negative repels negative, positive repels positive, right? Or if I make the mask positively charged, then the virus gets trapped in these fibers. So in this particular mask made by people at the Hong Kong Polytech University, they've actually charged the fibers, and the fibers in the mask will retain the charge for up to 3 months. And after that, you'll still have the mask protection from the particles, but it won't have the electrical charge on it. And then at Indiana University, all these little dots here are actually little nano batteries, microcell batteries that give the mask an overall charge, not enough to give you a shock, but enough to repel the virus. So, lots of innovation going on here. And, a little bit through here, I'm just sort of going to expose you to that, no pun intended. Okay. All right. Let us remember the droplets land on surfaces and they release their particles. So moving forward, and as you go out into the community, you want to think about this, right? Surfaces include things like doorknobs, light switches, the sidewalk, the floors, the handles of carts, even the food that you pick up in the grocery store, right? So, early on when I went to Giant a couple of weeks ago, someone was not wearing a mask. That individual coughed in their hand and then began picking up cans on the shelf to look at the expiration dates, and all the sneeze slime, all the cough slime that was in their hand has now been deposited on all those cans, all right? So the question becomes, how long can COVID remain viable on a surface? And what that means is, how long can COVID-19 be on the surface and still be able to infect you? So scientists did a study on this. This is the New England Journal of Medicine, and they looked at different materials. So this is published materials. The actual survival time of the virus depends on things like the temperature, the humidity, and the sunlight, because viruses are made up of proteins, and proteins are not stable to heat, right? So this is why people are hoping that as we move into the summer, the heat of the summer and the additional sunlight may kill virus particles that are floating around in the air and that have been deposited on surfaces. And I wonder myself if one of the prevention strategies might be that after classes are done, we just crank the heat up in the buildings overnight to as high a temperature as we can get to try to inactivate some of the virus and then turn it back down before students come in for the next day. So these graphs are kind of cool. I just wanted to show you there's some data here. This is our data science, our data analysis. But practically speaking, here's what this means, all right? How long can the COVID last on different surfaces, right? So notebook and tissue paper, 3 hours. Cardboard, your Amazon deliveries, 24 hours. Clothing and towels, 2 days. So think about this when you're walking outside and somebody coughs on you. If you're at the store or something, it gets on your shirt. The virus is going to be on your shirt for several days. So for me, when I go out, I always put all my stuff in the laundry as soon as I come in the house to get fresh clothes on. Stainless steel 2 to 3 days, plastic and glass 3 to 4 days. Your surgical mask, 7 days. So it's going to depend on the material. These times are not set in stone. If you look at different studies will say that some things can last up to 10 days and some things last only a few minutes. But this seems to be the most common consensus of the times that the virus will last on different surfaces. So again, something that you sort of need to be aware of moving forward. All right, so cleaning surfaces. When you clean a surface, You need to wear your PPE because you don't want to get the virus on your surface on you. So this would be a mask, this would be a glove, and preferably some kind of disposable covering for your clothing. You need to have an effective cleaning agent, right? So I thought about recommending some, but then I thought, no, I'm not going to do that. But you can look up cleaning agents on the CDC and the EPA, and the CDC also recommends how to clean and disinfect. One thing that people forget when they do cleaning agents is the contact time, right? This is not you just spray the cleaning agent on the surface and then wipe it away. Different cleaning agents should sit on the surface for certain amounts of time to be able to kill the virus. This is one of the main causes of infections in hospitals, is that people they spray their Lysol on the surface and then they immediately wipe it up. And it should have a contact time. It should be sitting there for several minutes. And the cleaning agent, the label on the cleaning agent will tell you how long to allow the contact. And then there are wiping patterns. And so if you want to learn the wiping patterns, take our BIOT 120 cell culture class. There are actually very specific patterns to clean with. And we always tease people to not be a bartender. If you ever watch the bartenders clean the bar, they wipe around in circles, and all that really does is just spread the contamination everywhere. We wipe in linear one-way patterns. Try this at home. You'll clean your spills up a lot faster if you do that. So with that in mind, I wanted to just take a minute and give a big shout out to our facilities folks. Thank you guys for the cleaning that you do, and thank you security for keeping us safe, right? I was a janitor for a couple of years, and I know how underappreciated those people can be, so say thank you to them. They're important. So our summary, routes of exposure. We have an infected individual. They cough without wearing a mask. Droplets travel in the air. They can form the aerosol nuclei. You can inhale that, all right? They can go onto a contact, onto a surface, and you can pick it up from the surface. And of course, I guess we're never going to shake hands again. So, and maybe not even fist bump, all right? But it's hard to be aware of this, right? We're probably really cautious the first 2 weeks, but as time goes by, we let our guard down. So you have to remind each other to keep up these precautions. Okay, so how do we prevent COVID distant transmission? Physical distance, barriers, face mask, and now hopefully I've convinced you if you can do it, wear gloves, right? If you can't use the disposable gloves, then rubber gloves that you use for dishwashing are fine. And guess what? You can reuse those and save the environment, all right? You can wash those. Don't touch your face. The face masks and the gloves are a great reminder to not touch your face. Keep your hands clean and wash with soap and water, or use hand sanitizer, but make sure that you get approved hand sanitizer, which has the right % alcohol in it. Lots of people are making counterfeit sanitizers right now, okay? So soap actually dissolves the membrane of the virus, the envelope of the virus. That causes all the proteins on the virus, which allow it to infect your cell, to be dissolved and go away. The virus loses its integrity. That 20-second wash with hot water and soap is what people have determined experimentally to be enough to, one, remove the virus and dirt from your hands, and also to begin to dissolve the envelope of the virus. I would also remind all of us to dispose of gloves and masks properly, all right? The number of gloves and masks sitting in the parking lots on the roadside, and I go hiking a lot. I now go to my secret trails because there's too many people on the main trails, but you see gloves on the Sino Canal and every place else. Take your gloves home, all right? And barring all of that, stay home. The last resort to the spread is the quarantine, the lockdown, okay? And that's what we're in right now, and hopefully we're coming out of. Okay, so if you don't properly protect yourself, you can get infected, all right? So symptoms of COVID-19 can appear anywhere from 2 to 12 days after you're exposed. That's why we want to keep you quarantined for up to 14 days if you think you've been exposed to the virus. Most symptoms appear within 4 to 5 days, but it may take 11 to 14 days for symptoms to appear in some people, right? The symptoms, we all know these: fever, headache, shortness of breath, coughs, runny nose. It's the same thing like a normal cold. But one of the hallmarks of COVID is that you begin to have trouble breathing, right? And the breathing can accelerate. Sometimes it goes away on its own. Sometimes it doesn't. We'll explain why that happens in the next couple of slides, all right? So some people, we call the people who are infected infecteds. This comes from a mathematical model. I know Dr. Rai would love me to put up a mathematical model, but I kind of avoided that. But there are models that are very evolved for this, and maybe Sanjay can actually give us a talk on that at another time. Okay, all right. But lots of people who are infected display no symptoms to mild to moderate symptoms. Okay, but when you are infected, the virus attacks virtually all of your organs, right? But especially your lungs, your intestines, your kidneys, and your heart. Okay, so I'm not going to go through the slide, but there's plenty of these slides on the website for you to figure out whether or not you have the cold, the flu, or the coronavirus. All right, and if you think you have it, you need to consult a doctor. But it's better to do it by the telephone to start with, as opposed to going into the clinic where there could be lots of other infected people, and there may not be room for social distancing. Okay, all right, now we're going to get a little more into the science here, how COVID actually enters your cells and replicates. You guys, all of us, are made up of cells. So this is my little schematic for a cell, and you're going to start to see how I envision things in life. I kind of look at things a little differently, but when I look at a cell, I go, we are all made up of cells. So basically, we're all covered with cells, okay? The virus is able to infect your cells, all right? So when somebody coughs at you, the virus goes down your airway, down your throat, and then into your lungs, all right? Different viruses find different ways to enter your cell and to take it over. COVID-19 uses a protein called ACE2, right? Angiotensin converting enzyme, ACE2. This is the protein that COVID latches onto, binds to, or the word is docks, just like the space shuttle docks with the space station. COVID docks with a protein that's on the cells in your lung called ACE2. Okay, so if we look at the different proteins on the surface of COVID, we have the N protein, which I haven't shown you, but that's inside coding the RNA. The M protein is the membrane protein. This helps the virus to assemble itself inside your cell and holds it together. The E protein also helps the virus to assemble itself and to leave the cell. But the big protein that everybody is concerned with is the S protein. This is called the spike protein on the coronavirus. And these are the red guys here in this model. And in the S protein, this is what docks with, binds to the ACE2 enzyme on the surface of your lung cells. Okay, so here's a lung cell. I've made a little picture of this is actually one of my better, by the way, homemade diagrams here, but this is a picture of the membrane of a lung cell, right? Here's our ACE2 protein. Remember, every protein has a specific shape. So through the process of evolution, the COVID virus came in and decided that it can fit into the ACE2 receptor. Once the virus has bound to the ACE2 receptor, once the docking has occurred, this now goes inside your lung cell. The virus takes over, and we'll show you that in a minute, and begins to replicate. This is a model that somebody who's a professional at making models has made, and this is actually made from mathematical modeling. So this up here is actually the virus. The red protein is the S protein, and this is the ACE2 protein on your lung cell. And why these are important is these are actually made from mathematical modeling of the structure of the two proteins, right? And when I have a good mathematical model that allows me to see the shape and the electrical properties of all the proteins, then I can interrogate that protein structure and let it inform me of potential drug targets. So by knowing the structure and having this mathematically modeled, I can again interrogate the structure. I can look at the different features of the structure, and those features may give me some information about how to create drugs to block the virus from entering into the cell. Okay. Now, I know you guys are rolling your eyes at this. I'm not going to go through this in great detail, but this is how COVID comes in, enters your cells, and takes it over. So I've just kind of shown you a little pathway. We're going to start up here at the left-hand corner. Here's our COVID-19. It docks with the ACE2 receptor. It's internalized, all right? Once the virus is internalized, it comes apart, all right? And then it goes through and begins to make copies of all its genetic material. The genetic material is then translated, remember we use that term, into proteins. The virus reassembles, and then it is emitted from the cell. and it goes on to infect other cells. All right, so you guys can relax now. That's as detailed as I'm going to get with this, but there are plenty of information on the website. Okay, all right. Now, why do we care about all this? Scientists spend a long time studying this because this helps us to identify potential drug therapy candidates and vaccine candidates. So understanding how the virus enters the cell, how the virus takes over the cell, how the virus exits the cell, by knowing each of those steps, it may allow us to create a drug that can prevent that and stop the virus in its tracks, or it may give us an idea of how to create a vaccine. Okay, so knowing this and knowing what the virus is, the question now becomes, how do we know if you're infected or not? So the current way that we do testing is we look for the RNA in the virus. Every organism has its own unique sequence of nucleotides, those A's, G's, T's, and C's, or in the virus case, it's A, U, G, and C, the nucleotides. So we're looking for the presence of that genetic material. So it starts by doing a swab, a throat or a nose swab. So if you are infected, the secretions in your nose, the saliva in your throat, will contain some of the COVID particles. So we're going to go in, take a cotton swab, collect it, and put it out. COVID-specific sequences of the RNA then are copied using a method called PCR, the polymerase chain reaction. All right. All PCR does is just copy the same piece of RNA over and over again to create billions of copies that can be detected. All right. Now, these are companies in Montgomery County that do COVID testing. GeneDx, which we actually, Montgomery College, has loaned some of our biosafety cabinets to use as part of the testing process, and I'll explain that in the next slide. Right on the Germantown campus, Zalgen Labs is trying to develop a rapid testing method. QIAGEN down the street on 118, and Thermo Fisher, and I always have to remember that Thermo Fisher just bought QIAGEN, so it's now Thermo Fisher QIAGEN. These guys make kits, and then there are specific test locations. This is a link to the testing locations in Montgomery County. There's more than I thought, but there are a lot of test locations available. At the moment, though, I believe that you need to have a doctor's note in order to get tested for COVID. That is not just walk-in testing for anybody right now. And I'm always a little nervous about that because as I watch all the testing done on the news, these guys walk up with their throat swabs and they have their PPE and their gloves on. They put the little cotton swab up your nose, you know, pull it around, put it into a tube, and then put that in a box. And then the next person pulls up, but they have the same gloves and the same PPE on to test the next person. So just something to keep in mind. You know, you got to think about things like this. Okay. All right. So how do we really detect COVID? Here's the virus. Okay. So we've collected the COVID on a swab. It's still infectious. All right. So we are going to do our work in something called a biological safety cabinet, which provides an aseptic environment. The virus is going to be contained inside of this unit. And you can see this person has the full-blown PPE on. All right. This is what we have loaned to GeneDx. So the first thing we do is we dissolve the virus to leave the RNA out and naked. So we now have naked COVID RNA, which is unique, right? We're going to get our test kit. I'm going to advertise for QIAGEN and Thermo Fisher because they've hired some of the biotech students, and we run the PCR reaction. So notice the test kit here has multiple components, okay? We're going to copy the viral RNA using something called a PCR thermocycler, again, which biology and biotechnology has lots of these. And we're going to copy this. And all we do is we copy the virus over and over again until after a couple of hours, we have billions of copies of the virus, and that gives us enough virus that we can actually detect it. We're able to see it with the aid of some equipment. All right. Now, challenges with the testing. First of all, contamination. All right. One of the reasons that we use the biological safety cabinet is that PCR is very sensitive. So if any virus particles from different patient samples are floating around, the virus from one patient can get into the tube from another patient, and then both people will test positive, even though one person only may be positive. We can have false positives, which says you're infected when you're not. And worse, we can have false negatives, which say that you're not infected when you really are. And if you've heard on the news lately, Abbott Labs, the testing that the president is using, they're having a lot of false positives and false negatives because their kit is not reliable. And Thermo Fisher actually on their website when they sell this kit, says negative results do not preclude the SARS infection, right? You still need to go to the doctor because a lot of these tests have been approved under emergency use authorization by the FDA, which brings us to some of the challenges in COVID testing. One is just the manufacturing of kits. So all of these different kits have multiple components, right? We have to make the chemicals that go into each of these vials. We have to fill the vials, we have to label them, and then we make lots and lots of these boxes. And remember, we didn't even know what to make to test for COVID 5 months ago. So think about that. 5 months ago, we had no idea how to test for COVID. There were no kits on the market. There were no chemicals known as reagents to do the testing with. Now we're making all these different kits, all right? But we also need to consider the supply chain. How do we package this? How do we ship it to the people who need to get it? Then we need to have test sites set up, right? And it's not just you do this in your basement, although some people think they can, but you need to have specific labs and the equipment to do that and the space to do it. And then we have what are called laboratory developed tests versus in vitro diagnostic tests. Laboratory developed tests are developed and run by a single lab, usually a hospital or university. These are not FDA approved tests. These are kind of like do-it-yourself tests, right? And you're hoping that people who do these are competent and able to do it. University of Arizona has taken this approach. They are charging their graduate labs and their graduate students and PhD researchers to do on-site testing, okay? And then in vitro diagnostics, all right, are approved by the FDA. And to get an in vitro diagnostic approved by the FDA, that can take 1 to 3 years before you meet all of the requirements to get FDA approval. So I kind of like this little meme that I saw. The whole country could be tested by midnight if Chick-fil-A was running these tests. If you've ever seen the lines at Chick-fil-A's, they get people through pretty quickly. So they're sort of a model of maybe how to do this. Okay. Part of the other problem with this is some of the first tests that were released by the CDC, the Center for Disease Control, they were contaminated, right? And so it's not necessarily a knock on the CDC, but CDC, NIH, they are academic institutions. They don't do things to the same standards that industry. They don't have to adhere to the same standards that industry does. And so this is sort of not unexpected because their job is to just produce research, so they don't need to adhere to all the rules and regulations that industry necessarily does. Okay, so let's go back for a second. The RNA test tells you if you are currently infected. The next test that people are looking at is the serum test, the blood test, and this is checking for whether or not you've made antibodies, all right? So if you have recovered from COVID, it means that your immune system did its job, it rose to the occasion, and it responded. And part of the immune response is to make antibodies, and antibodies are proteins that you make which detect and bind to the virus particles. And so the antibodies cover up the virus and prevent the virus from infecting your cells. So if I can detect the presence of those antibodies in your blood, I know, one, that you've already had COVID, that you've recovered from it, and that there's a chance that you may have some immunity to future infections, all right? So these are the tests that are coming up, a little bit longer to develop because the science and the production process for making antibody testing is a little bit trickier than making RNA testing. This is what Zalgen Labs and the Germantown incubator, the GIC, part of PICMC, is working on. Louis Branco, who's like one of the nicest people you'll ever meet, by the way. Really nice guy. All right. So this is a summary of the RNA tests versus the antibody tests, right? So the swab test tells you if you're contagious right now, the blood test does not, all right? With the swab test, we're taking a sample from your nose or mouth. The blood test is usually a finger stick, okay? And then the result is 1 to 7 days, depends on the backlog for the swab. For the RNA test, the antibody is hours to a few days, okay? All right, so I'm starting to run out of time, so I'm going to speed up here a little bit. How does your body respond to COVID? All right, your immune system is able to detect disease-causing entities such as COVID-19. We've already said this. Once your immune system detects that you've been infected, it rises to the occasion. All right. Initially, cells known as macrophages and neutrophils enter the scene. Right. So this is a little schematic. You have macrophages and neutrophils that are patrolling your body all the time. They just detect whether or not you've been infected. These cells release cytokines, which are chemical messengers. The cytokines recruit other immune cells and increase the rate at which the existing immune cells work, all right? So other immune cells, here are the cytokines, other immune cells, B cells and T cells get recruited to the site. They release more cytokines. Ideally, you kill the virus. But if you don't easily kill the virus, then you keep releasing more and more cytokines, and you get what's called a cytokine storm, all right? And so that's what COVID does. It overstimulates your immune system, right? And this results in massive inflammation, which causes tissue damage, because part of what cytokines do is they make your blood vessels leaky. So the cells lining your blood vessels come apart slightly, and the fluid in your blood starts to leak into your tissues, and you get swelling and inflammation, and it hurts, and your cells, your lung cells and other cells are filling with fluid, right? The cavities in your lung are filling with fluid, and they're also not getting the oxygen and nutrition that they need. Right now, interestingly, the ACE2, the same protein that lets COVID infect you, also degrades cytokines. So the ACE2 protein breaks down the cytokines that are causing the cytokine storm. And it turns out that young people and women especially have high levels of ACE2. So while it's easy for them to be infected, they don't get the same symptoms as elderly people because they have such a high level of ACE2 that they're breaking down the cytokines and not getting the cytokine storm. As you get older, the number of ACE2 proteins decreases, so you can still be infected, but you can't manage the cytokine production the same way that you can if you have lots of ACE2 receptors. So again, this is a target to alleviate the COVID symptoms. So how do we treat COVID? We do one approach is small molecule drugs. These are drugs that you can make chemically. The approach right now is to use small molecules that are already in use because we know how to make them and we know what their safety profile is. We know how they work, right? So what we're looking at is they are going to interfere with viral uptake, with the replication of the virus or the release. So remdesivir prevents copying of the viral RNA. Hydroxychloroquine, right, which I know everybody is in controversy, but hydroxychloroquine has been shown to prevent both the uptake and the release of the virus. The problem with all this data is people have not done controlled scientific studies. This is all sort of anecdotal studies. So doctors can prescribe drugs off-label if they think it will help the patient, but they're not really doing this in scientifically controlled studies where you can analyze the data and get a good statistical analysis. Then we have azithromycin, which blocks protein synthesis in bacteria, so I'm not really sure how that helps with COVID. Corticosteroids limit inflammation, and then people are trying combinations, which we refer to as drug cocktails. There are over 30 small molecules being tested against COVID. Plasma therapy is where we take the blood from people who've recovered, which will have anti-COVID antibodies in them, and we administer that plasma to other people, and those antibodies will bind to the virus and prevent it from infecting you. Okay, neutralize. Sorry for going fast here, but I want to try to wrap this up. So vaccines, right? Vaccines are designed to activate your immune system without making you sick. They create memory immune cells, so you have thousands of immune cells to respond to when you get really infected with the virus, and they hopefully hang out in your body for years, right? So there's lots of stuff that you can use to make vaccines. Right? We can use live bacteria or viruses, inactivated bacteria or viruses, parts of bacteria or viruses to make this. So this is what we're looking at with COVID. All right. Can we just kill COVID and inject it to you and stimulate your immune response? Can we take a piece of COVID, like the S protein that allows it to infect you, and give you just the S protein and raise an immune response to it? All right. So these are the different approaches that people are taking. And I'm almost on my last slide. Yay, guys, you've made it. Okay. The one I'm going to focus on is the RNA vaccine that was in the news yesterday. This was made by a company called Moderna, which is up in Boston. And I actually had a couple of students who went to Boston and were working for Moderna. One is still there, one has since come back. But they are funded by BARDA and by the Institute of Allergy and Infectious Diseases. This is an RNA vaccine, which is a novel vaccine. So the way that this works is you inject RNA, here's our RNA right here, into the patient. This RNA contains the information to make the S, the spike protein on the coronavirus. So the RNA is transcribed, it makes the S protein, and you make an immune response to that, all right? And now that the S protein is covered with antibodies, it can't infect your cells. And as of yesterday, they released the results of a phase 1 trial, which is really just testing for safety, but everybody who got this RNA vaccine, they were able to detect antibodies in their blood, which is what sent the stock market soaring, because you can make the RNA vaccine the same way that you make chemicals like aspirin and purify it. So you could make literally millions and millions of doses of this RNA vaccine in a very short time, and you can get it chemically pure. What we don't know is how long this is going to last. All right. So all of this goes to, as we say, lowering the curve. All right. And so this is really what we're looking at to reopen everything when there's so few people infected that the virus is less likely to be transmitted, and that we have a way to manage the people who do get sick. All right. So companies in Montgomery County that are working on vaccines are involved in COVID. Right. Novavax, Emergent Biosolutions, GlaxoSmith Kline, Astra Zeneca, Altimmune Scenario, Integrated Biotherapeutics, Vigene, and then there are other contributors: Bioreliance, Next Molecular Analytics, ACell, the United States Pharmacopoeia, NIH, and NIST. Everybody with an asterisk has hired students from the Biotech Program. All right. So I'm going to take a minute at the end here to shamelessly promote the Biotech Program, because we need students. All right. So the Biotech Program, you guys knew I was going to do this if you let me talk. So, but this is our last slide. So the Biotech Program prepares students for entry-level jobs in the biotech industry. We have placed in the last 15 years over 300 students at companies. For a 2-year degree, students start at $45,000 to $50,000 per year, plus benefits. Most companies will contribute $10,000 a year towards tuition through a master's degree. And there are right now, even before COVID, hundreds of job openings. The Biotech Program transfers to lots of local universities. If you're interested, if you have students who you think this might apply to, we don't need A students, we just need students who can do lab work and who show up on time and things like that. Contact Dr. Kelman or myself, or if you have additional questions about the presentation, feel free to contact me. And if you know people who want advanced training, we also offer BioTrain and BioTrack at the college. Mike Gove and Mark Nardone run these programs. BioTrain is designed for incumbent workers. BioTrack is designed for PhD level researchers. All right, I made it only 6 minutes over. That's pretty good for me. So thank you guys very much. I hope you found this useful, and I'm happy to entertain any questions right now. Thank you again. Stay safe, everybody.
Well, thank you, Dr. Jones. Wow, based on the chat box, this presentation was very, very popular. So we appreciate it. This brings us officially now to the formal Q&A segment of the presentation, and I have been keeping track of the numerous questions that have been coming through. So I'm doing my best to try to ask you these things in the given time that we have left. But I do want everybody here to know that we are going to review the chat log, and questions that we do not get a chance to ask and answer here in this space, we will answer, we'll direct it to Dr. Jones, and Dr. Jones can answer those remaining questions. All right. So
after I finish finals.
Absolutely. Of course. Of course. So I've grouped the questions kind of as best I can throughout this presentation by category. So starting with questions that were asked in the very early portion of the presentation about consuming meat. So one overall question is, can eating the meat of COVID-infected animals also infect that person who's consuming the meat?
Okay. So I'm going to preface this by saying I'm not an expert, but the reason that you cook your food is to inactivate all of the pathogens, all of the disease-causing organisms, right? So if you just went to the store and bought hamburger and didn't cook it, there is on every meat product you buy, salad, vegetables, there's what's called a bioburden. And the bioburden is all the unwanted biological material, things like bacteria and viruses that are naturally floating around in the air that land on those surfaces. So to COVID, I'm not sure what studies have been done, but my educated guess is that if you cook your meat properly, it will not affect you. I will say I love sushi. I have not eaten sushi since this started. Okay. So, but this is the USDA has done studies to say this is why you cook your chicken at this temperature for this amount of time, or your pork at this temperature for this amount of time, because they know what bacteria are most likely to be present on that particular type of meat. And they've done studies as to the correct time and temperature to inactivate those disease-causing bacteria. So hopefully that helps. That's a great question. I love these questions.
Okay, great. Thank you so much. Moving on to the next category of questions. So the next one is about community transmission. So here's your first one. If COVID lands on our surface, like our hands, but is not inhaled, can the virus still enter through the skin?
only if you have an open wound. Your skin is actually part of your immune system. And on your skin, there's two things. One, believe it or not, your skin is coated with bacteria. It's helpful bacteria, but the average human has 5,000 to 10,000 bacteria per square centimeter on your skin. Even if you take a shower every day, it's still there. So kind of between the skin and those bacteria, it provides a barrier. And then you also release through your skin enzymes that will kill the virus. So it should not transmit to you through your skin unless the integrity of your skin is compromised with an open wound.
Okay.
I still wear gloves.
Okay, well, that brings us to the next question, actually. So what happens if someone is just talking to us, not sneezing, but we're less than 6 feet away?
There is the possibility that you could be infected if neither party is wearing a mask, because you're still emitting droplets through normal talk. If you remember in the beginning, just talking emits 1,000 to 3,000 droplets, which I didn't know till I did this talk. So thank you guys for asking me to do this. So the dose is not going to be as high as a sneeze, but you have to think about this. The longer you're talking to that person, the higher the dose you're getting. Does that make sense to you guys?
Yeah, good.
Okay.
And then here's another community transmission type of question, but now related to the school setting. So what would be the ideal conditions to teach in a traditional classroom environment?
You know, I've thought about that. And I'm going to start by saying that's way above my pay grade. That's the reason I like to remain a professor. You know, I truly miss the face-to-face contact. In the size of our classrooms, I'm not sure how we do physical distancing. I can't imagine installing plexiglass in between every chair. And even if you do, you're going to have to clean it in between classes, right? I mean, people don't think about that, but when you go to the supermarket and you're talking at the teller with a plexiglass in front of them, they don't come on and clean that in between each customer, right? So basically, if you go at the end of the day, you're basically facing all of the COVID and whatever else people have emitted on that plexiglass. So you guys can sort of see how I think. You wonder anything, but I trust my immune system. So I would say for me, when we go back, I would like to be wearing a mask and I would like to be wearing gloves. And I probably will change my clothes, put on new pants. I put on new shirt every day, but I probably will change pants and shirt every day that I teach and wash. And, you know, my question for this, which is going to be an administrator question, is how do you deal with the people who don't comply? The students who don't want to wear a mask, right? Who don't want to social distance. So this is why I'm happy to be a professor, and good luck, you guys.
So the next category of questions are related to the masks when you presented on that earlier. What if you make your own mask with the cotton fabric with multiple layers? And then is there a suggested number of layers?
I don't know if there's a suggested number of layers, but multiple layers help, especially if you sort of put them at perpendicular angles. So, you know, one this way and then one that way, right? Because what you're trying to do, think of this as a screen. And if you put like a window screen, if you take two window screens and put them perpendicular to each other, then you make the holes smaller, right? So but you still have to remember to be able to breathe. And if you're using especially these kind of cotton masks and things like that, you want to be able to wash them. So you should make several masks and rotate them. So I had 5 N95 masks at home. I take them when I go hiking because out west there's forest fires and I sometimes need to wear them to protect me from the smoke particles. So I've been rotating those on a daily basis. I go out maybe once every 5 to 8 days.
Thank you for saying that, because that actually was going to be the next question. How long should we set the N95 mask aside before having to reuse it? So you're saying 5 to 8 days.
Yeah, well, I would say 3 days and put it in the sunlight. But the problem is if you put it in the sunlight can degrade the elastic bands. And so they will break more easily.
Okay. And what about the risk of contraction through the eyes?
That is, as far as my understanding is, again, I need to preface this by saying I'm not a doctor, I'm not an expert, but my understanding is that you can actually get viruses through your eyes. So I actually now wear, you know, I learned long ago that most people don't remember you, they'll never see you again. So I was wearing gloves at the store over 5 weeks ago, And now I wear not only the face mask, but I wear my sunglasses or safety glasses when I go shopping now, just to keep stuff out of my eyes.
Okay. And can we reuse, or not reuse, but wash gloves and masks for reuse? And if so, what's the best detergent?
I think any detergent works. I would not wash a cloth mask or an N95 mask. That will degrade the particles. A paper mask, you can certainly wash a cloth mask, sorry. But heat and detergent works. Gloves, you can wash, but make sure you check them for rips. Right? Anytime you're wearing gloves, you need to always make sure that you check them for rips. If you spray them with alcohol, and that includes hand sanitizer, that will begin to weaken the latex or the nitrile in the gloves, so they're going to be more susceptible to ripping. But you can sort of reuse them. But for me, no, I just have it. It looks weird, but if I run out of gloves, I had gloves for painting and stuff. I have these big black dishwashing gloves that I wear that go halfway up my sleeve. So you can just wear those and wash those and reuse those for a long time.
And someone just asked a question that is related to what we're talking about. Does a microwave oven kill the virus?
Supposedly, yes. But if you put your mask in there, it's going to melt parts of the plastic on the mask. And the same thing with your nitrile gloves. Okay. So what's not available right now, which I wish I had bought, but I didn't think I had enough, is when this is over and things calm down, you can actually buy handheld UV lights. Make sure you read the specs on it, but you can wave the UV light over your mail and stuff like that. Sorry. Right now I put my mail out in the sun for a couple of days.
Okay. And I have a handful of questions, which I will try to get through. We only have 15 minutes left, and I want to respect everyone's time. So if you can answer just a couple more questions, Dr. Jones. Earlier, you talked about hand sanitizers. What is the appropriate percentage of alcohol in hand sanitizers to be effective?
My understanding is 60 to 70%. But that means you can't make hand sanitizer by buying 70% alcohol at the store and then mixing it with the gel, because that will dilute it to below 70%. And the 70% range is better than the 100% range, because the water in the alcohol allows it to enter the envelope of the virus more readily than the 100% alcohol. So this is all stuff that people have worked out in the studies.
Okay. What about in cars with respect to air conditioning and heat? Are there precautions we should take?
That's a good question. Basically, don't get it in the car. And, for me, I actually now keep the windows rolled up because I see too many people as the summer is coming, sitting next to you when you're at a stoplight with the windows rolled down and not wearing a mask. But I do, if I mess up and don't take my gloves off before I enter the car or something like that, then I do wipe down the car. But again, just letting your car sit for a couple of days in the bright sun, you'll build up the heat in there, and the sunlight should kill things. That's my understanding.
Okay. All right. So here's the next category of questions. It's related to how we can move forward with this. How long will it take to develop a vaccine? I know we don't have a crystal ball, but what do you think?
No, we don't. I mean, it's interesting. We may have, they're going to be called vaccine candidates that will trigger an immune response. The Moderna one looks, to me, it's amazing that they've already gotten this result within 6 months. That's just unprecedented. If that works, then you could literally make millions of doses by December. But the reason that it takes so long to make any of these drugs is the safety testing. So what most people don't know is that you may discover a drug in year 1, but the clinical trials last 6 to 8 years to prove that it's safe. So you usually do 1 to 3 years of preclinical testing, which is in animals, lab animals, and then you do phase 1, phase 2, phase 3 clinical trials. Each of those lasts for 2 years. And then the drug gets approved. So it normally is a 10-year process. Mm-hmm. Right? You can get accelerated approval in times of emergency, but then you're not as sure about the safety.
Okay.
So I dodged that pretty good. Sorry, guys, but that's sort of the best I can say for this right now. I mean, it's a risk-benefit analysis.
Right. We appreciate that. Just one more question related to testing. How is it that some other countries have these drive-through testing for COVID do they just get the results real quickly, or is it that you're just collecting samples to then later test?
My understanding is they are collecting samples. They get sent to the lab, and then the results come back out. And, and again, so this is, you have to think about this. This is a lot of data to handle. These are a lot of samples to handle to make sure you don't mix them up, to make sure you don't cross-contaminate. So, but that is my understanding. Now, the antibody test should be, there are some tests that I see that I didn't include in here that they're trying to develop tests that give you a result in 15 minutes, sort of like a home pregnancy test. It's going to be modeled after the same sort of test kit. And you may even be able to buy those off the shelf. They're called OTCs, over-the-counter testing. So you can just take it at home. But, again, think about this. 5 months we have tests. That's just remarkable in my mind, knowing how the industry normally works.
Well, Dr. Jones, I'm going to ask you one more question. And then after this question, we apologize again that we can't get through all the questions, but we will look through them through the chat log and try to address those at a later date. But this is the very last question for you, Dr. Jones. Okay. Can you say a bit more about the COVID-prompted inflammatory disease that we have seen in children?
I can't. I don't really know enough about this. I will say this. What's been interesting now is there's some theories going around that the way to treat COVID, and whether it's an adult or a child, is to really focus more on the inflammation, and that the ventilators may not actually be necessary. They may actually be more of a problem, creating more of a problem than not. And, I'm not a doctor, so I don't know this, but in my joke group, which is where I got some of the joke pictures, we also spend a lot of time talking about COVID in there because they're all scientists who have worked either at biotech companies or at NIH or CDC. And we're having a discussion right now about is using anti-inflammatories to treat COVID another sort of course of treatment. But that is not doctor's advice. Sorry, I'm nervous about people, it's just a hypothesis. This needs to be really vetted by professionals under clinical trials. But it is interesting, I guess. What's a little scary to me is how different aspects of the disease keep popping up over time. That it's not just a straight symptomology that we see all the way through. So that's the best I can do on that.
No, and we're very, very grateful for that. This has just been an amazing wealth of information that you've provided us in the last hour or so. Dr. Jones, do you have any concluding remarks? I know you've spoken a lot, so do you have any last words for us? Thank you all for attending. Stay safe, and I really hope that some of this information was useful to you guys. I know I went through it fast. I probably melted your brains, but that's what I do when I lecture. So, and if you email me questions, just be patient. I'll get to them, but I am administratively challenged, so it may take me several days to get back to you. And don't hesitate to ask again. And thank you again. Thanks for this opportunity. Thank you for taking it. The recording and the slides will be available on the Summer Professional Development Institute website, which we shared the URL on the chat box. I'm going to turn this over now to Dr. Mills, if you have any concluding remarks that you'd like to share with us.
Sure. Thanks, Shinta. And Collins, wow, just absolutely amazing. I can see why your students are so enthused when they're taking your class, and why industry is so excited when they're working with you. Your passion for this is remarkable, and we can't thank you enough for giving this presentation. I do want to take a minute to let Dr. Rai just say a few words, and thank you. Sanjay?
Yeah, thank you, Mike. Collins, again, thank you very much. What an outstanding presentation. When we were planning this professional week, Mike, Shinta, and myself, we thought about, drifting a little bit from the routine type of professional week. So much information is out there about COVID-19. And we thought, we have a wonderful expert right here at the college. And especially you have this amazing ability of bringing scientific information and making it accessible to general audience. And this is what you did wonderfully today. Very, very helpful. We can't thank you enough for doing this. I remember I wrote to you and a few others on a Saturday, and you immediately agreed. And you put a lot of time into this presentation as well. It's very, very clear. It's not an easy thing to do. And for all the colleagues who attended the presentation today, I want to thank you for taking your time and giving your time to this professional development activity. The entire series so far is very successful. I want to thank Dr. Mills, Shinta, and others who are involved in the planning. Between yesterday and today, we had almost 600 attendees. I think it's wonderful. And, we have struggled with some of our own decisions at the college about when should we come back to campus. As you know, we have decided that our entire summer will be in the remote format. We're not coming to the campus. And we also made a very early decision, in fact, about fall of not coming to the campus and remaining in remote environment until things change significantly, whether vaccine or other things. And those were difficult decisions. And there were viewpoints on both sides, some saying, no, we should open, and some saying we should not. And after listening to Colin's presentation today, I am more assured that the decisions that we have made are good. Social distancing in our classes is extremely difficult. Some of our classrooms are very tight. 18, 20 students, social distancing and cleaning and enforcing masks and everything else. Those are challenges. We will work through this. As you have shown, Collins, some better technologies coming, better masks are coming, and those things will be a part of our decision making as we move forward. In terms of biotechnology program, as you know, I want to put a plug again, highly successful biotechnology program, serving all the major companies. And these companies are actively working on vaccine, antiviral drugs, and test kits. So as an institution, we are contributing to the issues that our society is facing at this time. Other than biotechnology program, we also have BioTrack, where advanced people with PhDs are coming. About 800 of them came to our Germantown campus just last year to get trained. Then BioTrain program. And I'm very happy to share with everyone that we are starting an early college in biotechnology from fall of 2021. So our biotechnology programs are expanding. And Dr. Jones is our leader, and we're very, very happy to thank him again. Thank you very much.
Thank you guys very much. Really appreciate the opportunity.
And I just want to reiterate to all three of you, Dr. Mills, Dr. Jones, and Dr. Rai, for having this opportunity to all of us here at Montgomery College. And Dr. Jones, again, we appreciate your sharing tips to keep ourselves healthy, to keep our families safe, and to keep our communities well. So thank you again. To everyone watching, as mentioned in the beginning, this presentation is the second of 4 presentations lined up this week. The Montgomery College Professional Week Speaker Series 2020 is designed to kick off the Summer Professional Development Institute. Please be sure to sign up on MC Learns to register and receive the Zoom link for the remaining 2 presentations. The next one is actually today at 1:30 p.m. We will be given by Dr. John Byers from UMGC. And then the last one will be tomorrow morning from Dr. Thomas Bailey, also from UMGC at 11 a.m. Thank you all again. We hope to see you all on Zoom. Thank you, stay healthy, and stay MC strong.